I've already mentioned at this point that not all integral membrane proteins are channels. Some can be classified as carriers or transporters. Those two terms, carrier and transporter, can be used interchangeably. Now, one example of this that we've already discussed is facilitated diffusion, which is a passive transport process that requires the help of a transporter. But from this diagram, you can see that transporters are also responsible for active transport as well. But before we get into that, I want to spend a little bit of time differentiating between transporters and channels. So just a minute ago, I mentioned that channels form a pore or a continuous path all the way through the membrane. But this actually doesn't happen with carriers. Carriers operate instead by something known as the alternating access model, which means there's never a continuous path all the way through the plasma membrane. Instead, a carrier will undergo a conformational change that opens up to one membrane face and allows the solutes that are in that compartment to bind to the carrier. It'll then close so that it's not accessible to the inside or the outside. That effectively traps the solutes inside, and that's known as the occlusion state. It'll then open it up to the opposite side to allow the solute to unbind and get released into the opposite compartment. Now, the other major difference is that solutes have to actually bind to transporters in order for that to occur, but they don't bind to channels. But transporters have a limited number of binding sites, and so that means that a typical carrier can only move between 1 and 5 molecules of solute per cycle, whereas a channel can move tens of thousands of molecules per second. But having binding sites can also limit the rate of transport. So in this diagram, you're looking at the concentration of solute on the x-axis and the rate of transport on the y-axis. And you can see that initially, as the concentration of solute increases, the rate of transport also increases. So for example, if you had a concentration of 1 solute, you could transport at 1 molecule per second. And if you had 2 molecules of solute, you could transport at 2 molecules per second. But eventually, you can see that the rate of transport plateaus. And that's a direct consequence of having a limited number of binding sites. So when I was in elementary school, I used to stay with my grandmother because she lived close to the school and I could walk, and my parents were working during the day. So because the school's on a busy street, we had to have a crossing guard. So keep in mind, for this example, this was back in the old days when you could hold the crossing guard's hand. But I had to hold her hand in order to cross the street. Now, most crossing guards have 2 hands, so if I was all by myself that day, I could hold the crossing guard's hand and we could cross the street, and she was able to transport 1 child per trip. But some days I would have a friend over after school, and then there would be 2 of us. So each of us could take one of the crossing guard's hands, and her rate of transport would increase to 2 kids per trip. But if a lot of kids were trying to cross the street, or I had a lot of friends coming over that day, she still only has 2 hands, 2 binding sites. So what ends up happening is that there's a queue that starts to develop because her rate of transport saturates. This happens with carriers and transporters, but not with channels. Now, from this slide, we can also see that carriers tend to be larger than ion channels, and that's probably due to all of those different conformational changes that they have to make that ion channels don't, open or closed. Carriers can also be classified based upon the number of solutes that they transport and the direction that they transport them. So a carrier that transports only a single solute is known as a uniport transporter. And if it transports more than 1 solute, then in order to classify it further, we have to know which direction the solutes are being transported. So if both solutes are being transported in the same direction, then that carrier is referred to as a symport carrier. But if the solutes are traveling in opposite directions, then that's known as an antiport carrier. We could also classify carriers based upon energy usage. So carriers that don't use energy work by facilitated diffusion, and carriers that do require energy for transport can be either primary or secondary active transporters. Primary active transporters typically contain an enzymatic domain that hydrolyzes and breaks down ATP. Secondary active transporters often use energy stored in electrochemical gradients. If you've taken other biology courses before and learned about free energy, then you could think of secondary active transporters as being driven by delta G. If that term isn't familiar, then don't worry, we'll be talking a lot more about it when we get to our membrane potential unit. Now, the two classification schemes, again, are not mutually exclusive, and there are some generalizations that we could make, but unfortunately, there's always going to be exceptions. So, for example, most primary active transporters are either uniporters or antiporters. And we can see that here with the sodium-potassium pump, which is the transporter that we're probably going to spend the most time talking about in our course. Sodium is highly concentrated outside the cell, and potassium is highly concentrated inside the cell. So this sodium-potassium transporter moves 3 molecules of sodium out and 2 molecules of potassium into the cell. Both of these molecules are moving in different directions, so we could classify the sodium-potassium pump as an antiporter. But because both of the molecules are also moving against their concentration gradients, it requires energy to work. So the sodium-potassium pump actually contains an enzymatic domain that breaks down ATP, and that gives it the energy it needs in order to drive that transport. So it could also be classified as a primary active transporter. Here we're seeing how the sodium-potassium pump can be plugged into that alternating access model. Again, open, closed, open. The other generalization that we could make, and of course there's always going to be exceptions, is that most secondary active transporters are also symporters. So you can see from this example, which is the sodium glucose transporter, it transports both sodium and glucose into the cell, makes it a symporter. But remember that bringing sodium into the cell is energetically favorable because it's moving down its concentration gradient. And that allows this transporter to use that energetic favorability to move glucose into the cell, even when it's energetically unfavorable. By coupling these two transport processes together, we get a sodium glucose transporter that works by secondary active transport. So here again is the sodium glucose transporter that's plugged into that alternating access model. Again, it opens to one side, it closes, opens to the opposite side. So I want to close by showing you an example of how passive and active transport can work together. So here we're looking at an epithelial cell in your gut that's responsible for glucose absorption. On the basolateral membrane down at the bottom, you can see that there's a sodium-potassium pump. We just talked about that. That pumps the potassium into the cell and the sodium out of the cell. And that helps to keep the sodium concentration inside the cell very, very low. If we come up to the apical membrane, the top membrane, you can see that the sodium glucose symporter is located there. That uses the concentration gradient for sodium to transport glucose. As glucose gets transported in, the glucose concentration inside the cell increases to be higher than the interstitial fluid, and that allows the glucose to get absorbed into your intestinal fluid down at the bottom through facilitated diffusion. So what we're seeing in this example is a situation where active transporters help to set and maintain the concentration gradients that the passive transporters require in order to work.